You prefer them like this, or do you prefer them the way they were before? Like that. Don't go to sleep. Okay, that's good. It's easier to see the screen, I guess. Okay, because we do have a few detailed slides this time. I haven't yet looked at the discussion board. I'll do that this afternoon, so if you put questions up there, I'll try and get them this afternoon. While people in the lab are beavering away on drills and things. Yeah, right. Anyway, so, yeah, not a very exciting thing to do. I also realized that I can talk all the way up to 1:45. Thank you for letting me know that. And so I will. So there's a mercilessly large set of slides to get through today. Now, I'm fully recovered from jet lag, and I know that. Hopefully we can move along. And do ask questions, right? Somebody really came up with some really good questions at the end of the lecture to explain things, and that's fantastic. If you can think of those kinds of questions during the lecture, that's even better. So do stop me and ask questions. So, are there any questions about last time's material? Somebody sent me an email saying, when do I sort of put up any revisions to the slides or new slides? And the answer is, I'll try and do it the day after, if not by the afternoon of. But it might be busy after I do my lecture. I might go somewhere else, so it waits till next day. But I'll try and do that, all right? Okay, so we're going to continue. Now, I talked about the various methods that you can use to try and uncover the diversity of neuronal structures and learn and use them to distinguish one neuronal type from another. And we looked a bit about what methods you might use to look at sort of layers of neurons, distinguish layers of neurons in tissues. Now I want to talk a little bit about the functional methods that you use to look at neuronal activity. And also just sort of introduce to you the basic sort of terminology of the functional activity of neurons. So we talked about how neurons encode information in two ways, and one is through these graded potential changes that occur by and large in the dendrites of neurons, and they're little blips of membrane potential change whose height, amplitude, is graded with the intensity of whatever's stimulated. And then, if those graded potential changes in the dendrites reach a threshold level, then an action potential is triggered. Okay? And if the stimulus persists for long enough, then a series of action potentials will be produced. The graded potential change will stay high, and as a result, one action potential after another will be produced. And that the action potentials carry information in terms of their frequency, how many of them occur per second. So we're going to look at a concrete example now, very quickly, of a sort of complete neural circuit. So you can place these potential changes and the methods used to record them in context. And this means that we'll be looking at pieces of anatomy and neuronal anatomy that you haven't covered yet and that we'll get to in detail later on in the course. But we have to start somewhere, and so let's dive in with this now. So the basic unit of function in the nervous system is something that's called a neural circuit. And this is basically a daisy chain of neurons. And one neuron releases neurotransmitter onto the next one in the daisy chain and either stimulates it or, as we'll see, inhibits its activity. So this is sort of a very simple daisy chain. It's the reflex that occurs when you go to the doctor's office and the doctor taps the tendon on your leg and the bottom of your leg swings out, hopefully. It seems to quite happen for me. I always feel as though I'm going to be subject to some terrible further investigation because I wasn't sort of vigorous enough. And if you try and do it to yourself, it never ever works. So don't try and test your own reflexes. You'll conclude conclude that you're dead if you do. All right. Anyway, so the idea is that doctor comes along with this big rubber mallet and bashes the tendon that's basically connected to your kneecap, right? Just below your kneecap here, this one here. And what that does is it produces a very sharp, very short, and not painful, hopefully, stretch of this muscle here, right? So this is the extensor muscle. It's the muscle that pushes your leg out, right? So the muscle is stretched because when he bangs here, it causes the tendon to move in this direction, and so it stretches the muscle. And that stretch is picked up, the motion of the stretch is picked up by a sensory neuron. And now this sensory neuron, the terminology used to describe this sensory neuron is a little bit obscure. Sometimes it's called a sensory neuron. Sometimes it's called a sensory receptor neuron. And sometimes it's called simply a sensory receptor. And sometimes it's called a receptor neuron. Okay, so it's some combination of sensory, receptor, and maybe neuron. And so you need to think of this in context, because in sensory receptors, neurons that respond to things in the environment, like the muscle stretch, the sensory receptor cells, the neurons, have in their dendrites sensory receptor proteins. We'll look at this in detail. And those are also called sensory receptors sometimes, right? And so you just have to use the context to decide whether we're talking about a receptor neuron or a receptor protein. Okay, just use your common sense. All right, but here, this is a big picture. We're talking about receptor neurons or sensory neurons rather than the proteins. All right. So the hammer tap stretches the tendon, which in turn stretches sensory receptors. So the dendrites of this sensory receptor neuron are wrapped around muscle cells. So when the muscle cells, and these are special little muscle cells, when they're stretched, the sensory dendrites get distorted because they're wrapped around the muscle cells. And that distortion of the sensory dendrite results in the production of action potentials, as we'll see, which travel along this process here, this axon here, through the cell body, out along this axon, and into the central nervous system, into the spinal cord. So this part of the diagram represents a section through a little part of your spinal cord, right? At probably around this level here of the spinal cord, where the neurons are located that receive information and send information to the lower limbs. Okay, so the sensory neuron axon runs into the spinal cord through this structure here called the dorsal root of the spinal cord, and then it divides. Right, so the axon divides and sends two branches, and we'll follow them both. The first branch of the axon ends in a nerve terminal that makes synaptic contact onto what's called a motor neuron. So this is a neuron in the ventral part of the spinal cord, the lower part of the spinal cord, which, or the ventral part of the spinal cord, which it then has an axon, this motor neuron, has an axon that goes back to the extensor muscle. So this is called the extensor motor neuron. Right? The other branch of the sensory neuron makes synaptic contact with what's called an interneuron in the spinal cord. An interneuron is sort of a generic name for a neuron that doesn't contact a motor neuron or a gland, and is also not sensory in nature. It's just a neuron that's in the middle of the daisy chain. All right? So it doesn't really mean anything except that it's just... And of course the vast majority of neurons in your brain, in your nervous system, could be correctly described as interneurons, right? Since most of them are neither sensory nor motor. So this interneuron then contacts another motor neuron, and this is the motor neuron that muscle, the one that causes your leg to flex. All right, so there are several interesting things already about this circuit that are general principles. Right? The first is that axons branch. Right? There are branches. So one neuron, the sensory neuron, can contact many motor neurons. Right? We're just showing two of the branches here. The second thing concerns the sensory neuron. Remember I told you, I drew a picture of a bipolar neuron that has some kind of dendrites out here. There's dendrites. It has an axon, which may be myelinated. And then it has a break, and then down here we'll have a nerve terminal. Okay, so this is sort of the classic neuron, right? Dendrites, cell body, axon, and nerve terminal. But this guy here, the sensory neuron, it's not that like that. It's got a cell body. It's got two processes that go in either direction, one towards the spinal cord and one towards the muscle. And then out at this end are these sensory dendrites that wrap around the muscle cells. And out at that end is the nerve terminal in the spinal cord. And then actually both of these areas can be myelinated. Both of those projections can be myelinated. So this guy goes sort of dendrites, axon, cell body, axon, nerve terminal, if you want to put labels on this. And this just reminds you, along with what we discussed in the previous lecture, that neurons are very varied in their structure. Basically, they will adopt whatever structures allow them to perform their task, to transmit and process the information they're designed to transmit. They've evolved to transmit or process. So what's happening? We'll see what's happening physiologically in this structure in a moment. So this synapse here, there's more than this. This synapse here onto the extensor motor neuron is excitatory. Okay, what does that mean? It means that when action potentials occur in the sensory neuron, they drive the production of action potentials in the extensor motor neuron. Right? Excitatory. This is an excitatory synapse. This synapse here onto the interneuron is also excitatory. And so when action potentials are generated here, the interneuron becomes excitatory. Becomes excited as well, generates action potentials. Now, in terms of the motor neuron, this motor neuron is directly excited by the synapse. And so that's the reason why when you tap here, action potentials are generated in the sensory nerve, they're generated in the motor neuron, and the reflex arc produces a contraction of the flexor muscle. Oh, sorry, the extensor muscle. So you tap here, action potentials travel around, this cell at the motor neuron is excited, this guy contracts, and the knee jerks out. Alright? But the knee can't jerk out, really, unless the flexor muscle is relaxed. If the flexor muscle is still in some kind of slightly excited state, contracting state, it's not going to allow the leg to come forward. And so what has to happen is that this interneuron has to inhibit the production of action potentials in the flexor motor neuron. So this synapse here, this one here, is inhibitory. In other words, when neurotransmitter is released from that synapse, the frequency of action potentials in the flexor motor neuron is depressed, is reduced. Okay? And any ability of that flexor motor neuron to generate action potentials is suppressed. So that means that the flexor muscle relaxes and allows the extensor to drive the kick of the leg forward. Questions about that? Yeah?
Does that kind of work in the reverse order? So if the flexor muscle were to contract with the extensor, relax in the same fashion? With an interneuron?
You know, I don't know, I can't honestly give you the answer to that particular question. It just never comes up. Thank you. It's great. Put it on the discussion board and I'll try and find the answer, you know? But what I can say is that every muscle in your body has these sensory neurons in it. And that this loop here, this loop excitatory loop, certainly is present in almost every muscle. Every muscle that's involved in posture. Anyway, there are a few that don't have them. The muscles that control your rapid eye movements don't have these sensory neurons in them. So this feedback loop here is pretty much a universal building block of muscle physiology. Yeah? The inhibition that occurs on the antagonistic muscle, I'd say, yeah, it really must be also part of the building block. So my bet would be that this occurs in a reciprocal way. But I can't say that for certain because I've never actually been told that. Because nobody's ever asked the question. So thanks. There's another question. Yeah?
What's the clinical significance of this?
We'll talk about that when we talk about motor neuron. It basically it's simple. If you're holding a mug of beer, right, and somebody pours a beverage into it, right? Yeah, so they sneak up on you and just slop beverage into your mug. What's going to happen is your hand is going to start to fall, and you will be in terrible danger of losing the beer, right? So that's going to stretch. That's the stretch. Right, in this case, it's the flexor that gets stretched. Yeah? And then what will happen is this reflex will drive a contraction of the flexor in this case, which will bring your arm back up. And you won't lose the beer, right?
Sure.
Well, that's significant enough for me. Yeah, sorry.
Okay, but why do they do this when you go to visit a doctor? Why, what's the reason?
The reason is to check the connectivity of the links between inside the spinal cord, and if there's any degeneration of the spinal cord, or if, for instance, the sensory neurons are degenerating, which occurs in a number of neurodegenerative conditions, including significantly syphilis. All right, because syphilis was a huge scourge, and so this became part of the traditional armor, and syphilis results in degeneration of these sensory neurons. And so it was part of the basic armory that doctors had to diagnose syphilis. It's actually not that much used. I mean, a lot of these neurophysiological tests are not that much used, or they're used largely sort of pro forma now, but yeah, so that's what it's for. Probably multiple sclerosis would also show up. Okay. All right, so I think we've talked about... So the other principle here is that of inhibitory synapses. All right? So lots of sort of basic principles. Okay, so let's talk about what's happening in terms of the membrane potentials or the action potentials in these cells. So the dendrites of the sensory neuron are stretched. Okay, and this results in the opening of channels in the sensory neurons. So out here, in the sensory neuron dendrites, channels open when they're stretched, and largely sodium and maybe a bit of calcium comes into the cell through those channels. And that results in a positive increase in the membrane potential because they're positively charged ions. And that that is the cause of the generator potential, or the so-called graded potential change, which is called the generator potential, the sensory generator potential. So the graded potential change in the dendrites results in, the distortion of the dendrites results in the influx of sodium and calcium ions and this graded generator potential, right? Positive going. And if this reaches threshold, if the generator potential reaches threshold, if the stretch is significant enough, right, that enough sodium and calcium come in, and this reaches threshold, then an action potential will be generated, or a series of action potentials if it's a prolonged stretch. And so the stretch opens channels in the sensory neuron and is transduced. That's the name of this sort of game here, this process, transduction, is transduced into an increased action potential frequency within the sensory neuron. So this is where those graded potential change, one way in which those graded potential changes occur in the dendrites of neurons, because there are channels or some other mechanism, or there are channels that are activated by the sensory stimulus. Right. Then the action potential is transmitted along the axon. It invades the cell body briefly and then travels down the other side of the axon into the spinal cord. In the spinal cord, the nerve terminal releases neurotransmitter, and the branching ensures that neurotransmitter is released onto both the interneuron and the extensor motor neuron. As a result, the extensor motor neuron action potential frequency increases, so does the interneurons, and then the action potential travels along the motor neuron axon to synapse onto muscle cells, and the muscle cell contracts. All right, so that's the basic scheme. So if you give a stronger tap, then this will generate a higher frequency of action potentials, a brief burst of high frequency action potentials. If you give a strong tap there, you'll get a lot of sodium coming in. The membrane potential will stay above threshold for a while, and you get a little burst of action potentials. Then the higher frequency, a stronger tap produces higher action potential frequencies in the sensory neuron, hence in the motor neuron and in the interneuron, and will generate a stronger contraction of the motor neuron, of the extensor, and a stronger relaxation of the flexor. Okay. And then, as I mentioned before, the maximum rate of action potentials, if you really whack your knee, and I don't suggest you do that because you probably injure yourself before you get to the maximum, is, in all neurons, is about 1,000 action potentials per second. All right. Okay. Now, one of the things that you might have realized in talking about this inhibitory pathway here is that I mentioned that what happens is that when the interneuron is activated, it inhibits the flexor motor neuron, inhibits it. And that presupposes that in that flexor motor neuron, there's a steady sort of background level of action potentials, which is then suppressed. Yeah? So that the resting state of this neuron here must have a few action potentials per second, at least. Right? And so this brings us on to a general point, a very important general point about all neurons, is that most neurons, almost all of them, show a basal resting action potential rate. So even when the neuron is not excited, quote unquote, it will be, even if the function of the neuron is not being commanded, it will be producing from 1 action potential per second to maybe 10 or so action potentials per second. And some of neurons have basal action potential rates of hundreds of action potentials per second. Yes?
So, in the case of the flexor muscle, where did that go? Like the sensory neuron goes into the neuron, and then where did that action potential go into the neuron?
So, what happens here firstly, action potentials never themselves travel physically beyond the cell that they're in, right? Because they can't, as we discussed, I think in the first lecture, they can't cross the cell membrane directly. But neurotransmitter will be released here. Yeah?
Specific inhibitory neurotransmitter?
Yeah. Well, neurotransmitter will be released there, and what will the neurotransmitter will do is generate a negative going graded potential. And we'll come to that later. Yeah, and then that lowers the level of action potential, inhibits the production. Yeah? So when your leg is at rest here, right, it's just sort of hanging out, you can imagine that there is a certain level of activity, nevertheless, in both the flexor and the extensor motor neurons. And that level of activity is raised in one and lowered in the other when the reflex is activated. Yeah? And so this brings us to a very important point, that information in your nervous system can be conveyed by either an increase or a decrease in action potential frequency, or both. Obviously not in the same cell, but in this case in two different cells. So that's a very important point to make, right? So when you think of yourself at rest or asleep or not doing anything, there is still a tremendous amount of activity in the neurons in your brain. A lot of them are actually more active when they're at rest than when they're processing information, because the way they process information is by reflecting, it's reflected in inhibition of their activity. Okay. So how can we record this action potential activity? So the easiest way to measure the electrical activity of cells is to simply place a couple of wires outside the neuron. So you try and get two pieces of wire, metal wire usually, but sometimes little glass micro pipettes, and you just place them outside the neuron. And what they do is those extracellular electrodes, as they know them, pick up the currents that circulate around the neuron when it produces an action potential. And we're going to go through electrical circuits and electrical current in detail in another couple of lectures, but I just wanted to get this point clear. So if you put a wire here and a wire here, so you put two electrodes close to this axon. When the action potential comes by here, is the inside of the cell positive or negative with respect to the outside? When the action potential arrives, at the peak of the action potential, it's positive, right? And the resting membrane potential is negative, all right? So if the action potential has arrived at this point here, it's peaking at this point here in the neuron as it sweeps down the neuron. The inside of the cell is going to be positive here and negative here. Right? And that is driven, that change in potential, as you know, is driven by an influx of sodium at this point here. Right? But electrical current has to flow in circles. It has to be a complete circuit for electrical current. And so the current that's flowing in here, that's carried by the sodium ions at the peak, has to flow out somewhere else. Right? So you've got a circulating current that occurs as the action potential sweeps past. And it's this current that is picked up by the extracellular electrodes. Because as this current moves from this point to here, the outside of the cell, the fluid, the goop that surrounds the neurons, all that dreadful stuff that's in our bodies, right? The extracellular space constitutes an electrical resistance, right? And as you may know, when current flows through an electrical resistance, it causes a change in electrical potential across the resistance. And it's that change in electrical potential that is registered by the two electrodes. is captured by the two electrodes. Okay? So this is extracellular recording. It measures the current, picks up the currents that circulate around the neuron when it produces an action potential. And it picks up that current by virtue of the fact that when the current circulates between these two electrodes, it generates a potential change. Yeah? So there's a potential change. Now these potential changes are very tiny, and as I say, they don't reflect exactly the membrane potential. You're not measuring the membrane potential here. You're measuring the current that's flowing around the neurons. And so what you see electrically when you make these recordings is an upward blip of potential followed very shortly after by a downward blip of potential. That's because when the action potential reaches this point, the situation is reversed. Now this point is at rest, this one has the action potential, and the current's flowing in the other direction. And so you see this rapid upward and downward blip every time an action potential goes past. Right? And that's called an extracellular recording. And so here's our sensory neuron. And we've got an extracellular electrode somewhere on the axon here, and we see these action potentials. When the hammer taps, the action potential of the sensory neuron increases, the action potential of the motor neuron, the extensor motor neuron increases, the action potential of the interneuron increases, and the action potential frequency of the flexor motor neuron is reduced. Yeah? And you notice that in all cases, there is some level of activity prior to the stimulus, this basal level of activity. Okay, so that's one way of measuring the activity of neurons with extracellular electrodes. The second way that you can measure the activity of neurons is by puncturing the cell membrane with a little glass pipette. So you take a glass pipette, right, hollow piece of glass, and you pull it. You actually heat it up in the middle and you pull it out, so you get this tiny, tiny sort of needle, right, like a hypodermic needle but made of glass. And that needle will have its tip will be generally a fraction of a micron in diameter. And you push it through the cell membrane. And something magical then happens. Actually, the cell membrane seals around the glass. And so now you have a pipette that is sealed into the cell membrane. And the cell recovers, it tolerates in most cases this glass that's stuck into its membrane. And you then, if you fill this glass with an electrically conducting solution, then you now have an electrical contact between this wire here and the inside of the cell. And so you can now measure the electrical potential inside the cell relative to outside. You need another wire, which isn't shown here, another electrode, which isn't shown here, that's outside the cell. Okay. So now we're directly measuring this membrane potential here. And you can impale, it's called impaling, you can impale the sensory neuron, or any of the motor neurons or interneurons, in theory. Okay, so what do you see when you do that? First, a bit of terminology. When you talk about membrane potential, when the membrane potential goes more positive, it's called a depolarization. When it goes more negative, it's called a hyperpolarization. It's terminology. All right. Okay, so what you see, as we've talked about before, is you can see action potentials and graded potentials. Alright, those are the two kinds, and this just goes over their properties, and we've talked about that in plenty. So let's have a look at this in detail now. So here's an electrode that is impaling, an intracellular electrode that is impaling the sensory neuron. And there's actually a mistake in this figure. If you're going to get the kind of recordings that are shown down here, the electrode should actually be impaling the dendrites and not the cell body. Okay, so I realized this morning when I was going through the slides. It'll be in the final version of the slides. But the book, if this is from the book actually I think made an error here. So if you record from the dendrites of the neuron out here, then when you tap, if the tap is given at this point here, you'll see a relatively slow potential change, relatively slow, it still goes up in a few milliseconds, right here. And this is the graded potential that's caused by the opening of those sodium channels and calcium channels. And when that graded potential reaches the threshold level, it triggers a much more rapid action potential, which will rise all the way to plus 50 millivolts. All right? So that's what you see if you record out in the dendrites. If you record from the motor neuron, let's look at the extensor motor neuron here. No, if you record from the interneuron, let's look at the interneuron here. Then you'll see again, when you give the tap, firstly there'll be a delay. Right? Why is there a delay? Right? Because the action potential has got to get down the axon. All right? And it takes a few milliseconds for that to happen. So firstly there'll be a delay, and then you'll see a graded potential here that results from the action of neurotransmitter that is released by the interneuron released by the sensory neuron onto the interneuron. Okay, so that's the second way that graded potentials can be generated, as a result of neurotransmitter opening sodium or calcium changes, calcium ions or other ion conductances in the membrane of the postsynaptic cell, this one here. So this is an excitatory synapse. So the graded potential change caused by the neurotransmitter in this interneuron is positive going, depolarizing. And then again, if it exceeds threshold, you'll see an action potential superimposed upon it. If the tap is very small, then you would only see a little bit of a generated potential there. And if the sensory neuron didn't release enough neurotransmitter, you'd just see a little generated potential there. It wouldn't reach threshold. And so there'd be no response. Okay, now here's the extensor motor neuron. And this, again, is an excitatory synapse directly from the sensory neuron here. And again, you get neurotransmitter released from the terminal here onto the motor neuron. You get a graded potential change that's caused by the action of neurotransmitter-gated channels. And then you get the action potential coming off it. All right? The flexor is another story. The flexor motor neuron is a different story. Here, you activate the tap will generate a negative going inhibitory graded potential change. All right? And that's because the ions that flow through the neurotransmitter-gated channels are different. So you'll get a negative going change, not a positive going one. And we'll go into all of that in detail later. All right? So the reason why one synapse is excitatory and the other one is inhibitory is because of the direction of these graded potential changes that are caused by neurotransmitter release. Any questions about that? Yeah?
Just in general for procedure, is there any advantage over recording something intracellular versus outside intracellular?
Yeah, so if you want to know just whether a neuron is active in a particular circumstance, is it driven by a particular sensory stimulus, you would use preferably extracellular recording. Also, if it was a really teeny tiny neuron, you can't get the stupid bits of glass inside it, all right, without killing it. But if you wanted to know what is the biophysics, what are the mechanisms of the synapses, right? What ions go through the synaptic channels, you'd use the intracellular recording method. Okay. Yeah.
So the hyperpolarization is caused not because of Na or C channels opening, but because of channels.
Chloride, for instance, if chloride channels open, then chloride ions would go into the cell, it turns out. We'll talk about that in the mechanisms of all of that later. I'm just trying to get these ideas floating around so that when we meet them in earnest, you'll have some of the context there, all right? Okay, so these methods for recording from single cells can be extended to recording from living brains. But they're invasive, all right? In other words, you have to open up the skull in order to stick your electrodes in. So I just want to cover the sort of extension of these methods for recording from brains. So maybe this will answer some of the questions about when you use these. So extracellular electrode recordings of electrical activity produced by a single neuron located near the electrode are called single unit recordings. Unit meaning a single neuron. Because I think you can understand that if there's another neuron nearby here, another axon nearby, for instance, you're going to detect the currents to some extent that are flowing around that axon as well. And so it's actually quite tricky to get your electrodes close enough to a given cell to just measure the response of that cell. All right? And sometimes you have to use fairly sophisticated computer software to sort of deconvolute what you've recorded so that you can distinguish between what's coming from one neuron and what's coming from the other. Right? That's another advantage of intracellular recording. If you're recording intracellularly, you are unequivocally recording the electrical activity of that cell. Okay, but if you manage to do the extracellular recording trick on a single neuron, it's called a single unit recording. Everybody loves you. You can also measure intracellular electrode recordings from single neurons as well, I mentioned that. And then it's sort of unequivocal. The other technique that's much used has nothing to do with electrodes. It has to do with fluorescent dyes that monitor usually calcium concentrations inside cells. Because it turns out, as we've met already here, that when neurons are active, often calcium channels open in their plasma membranes. And so the activity of neurons is often accompanied by a rise in their intracellular calcium concentration. And so if you can get fluorescent dyes that fluoresce when they bind calcium into neurons, you can monitor indirectly their activity by monitoring their calcium concentration. So this is another method. And so this is somewhat less invasive, right? You don't have to stick things into treat your brain like a pincushion and stick electrodes into it. You can coat the surface of the brain or the surface of the brain slice with the fluorescent dyes. The cells will take up those dyes, and then you can use a fluorescence microscope to look at the cells glowing at you, as it were, fluorescing at you in that tissue. So you don't have to stick anything into it. All right. The other great advantage is that you can monitor the activity of hundreds of cells at once using the fluorescent dyes. You can also make large-scale arrays of extracellular electrodes and do the same thing. But the fluorescent dyes are probably marginally more convenient, easy. Yeah? Yes. Yeah, I mean, I don't know about the fluorescent dyes. I don't know that any of those are approved for clinical use. But you can certainly record extracellularly. And that's done routinely during brain surgery. If anybody's ever had brain surgery here, I won't ask, but you know, one of the problems with brain surgery is that everybody's brain is slightly different. So if they want to remove, say, a tumor from part of the brain, they often need to verify that, you know, what function is that part of the brain subserved, so they can figure out, you know, are you going to stop breathing if they remove that part of the brain, or to what extent, what sort of prognosis can they make for your future recovery? No, they don't. They treat the brain like a pincushion, or they put electrodes on the surface of the brain, usually. As far as I I know they're not used, but I might be completely wrong about that. Again, put it on the discussion board, and but my feeling is that they're not used. For some reason, I think it's because the people who do this work are surgeons, and they're sort of uncomfortable with the idea of applying chemicals. So do you think? Yeah. So do you know any differently?
Yeah, no, I'm just curious on like how is that be different than something like a functional MRI? You get what part of it?
So a functional MRI occurs in a huge instrument, and so as far as I know, there aren't. It's not a really good environment for a hugely expensive instrument. So it's not a great environment for doing surgery in and around.
My question was, if they know where the tumor is, why can't they do that to decide what that part is?
So they know where the tumor is, but what they don't know is whether supposing it's a tumor that's close to the neurons that control breathing, they don't know they might not know in any one individual how much of that tumor they can remove and still keep you breathing. Whereas if they monitor the activity of the neurons while you're breathing and determine which is the area of the brain that's responding or driving the breathing motion, they have a better chance. They can decide how much of that tumor they want to take out. All right, and because a surgeon doesn't want to take out the whole thing based on a previous MRI scan and then wind up with a dead patient. So that's an extreme example. Okay, I don't know whether that has ever occurred. It's an extreme example, but this principle applies nevertheless. And for situations where they're removing parts of the brain, for instance, to treat epilepsy, which they don't do so much anymore, they tend to use drugs, but they used to do it a lot, you don't want to get rid of somebody's ability to perceive a stimulus or to drive a particular motor part of their body, motor-wise, just because you want to reduce their number of epileptic fits. So that was my personal experience. I didn't have epilepsy. A cousin of mine up in Massachusetts had epilepsy, and so she was put under local anesthetic, and she was asked, to move different parts of her body, and they stimulated her vision and other senses so as to figure out, as they were removing parts of her brain, whether these were connected to anything particularly vital. She actually went in for surgery just before an general election, presidential election, and she voted absentee because she was worried that when she came out of the surgery, she might be a Republican. Apologize to Republicans. I guess I promise next time I will be a Democrat. Okay, so here's an example, right? So you take off the skull and you open it up and you stick a recording electrode in and you measure the activity of neurons in this part of the brain. And so what you can do with this is, for instance, to ask the question, if we're recording from making a single unit recording from a neuron in the cortex of the brain, that is in the part of the brain that deals with somatosensation, with the sensation of touch, then if we take a stylus and touch various parts of the animal's arm here, how does the neuron respond? And you can see in this case that if you touch in this area here, you get a vigorous excitation of the neuron. If you touch out here, nothing happens. And if you touch in this region here, you get actually an inhibition of the neuron. And so this neuron is said to have a receptive field, an area to which it is sensitive, and it has an excitatory center of its field and an inhibitory surround to its field. Again, information is being carried both by excitation and inhibition in this case. Both excitation and inhibition is carrying information about where the synapse is. And this turns out to be a very common sort of sensory way of arranging the sensory fields with these excitatory, inhibitory surrounds. Excitatory, sensory surrounds. So this is the fluorescent dye idea. Here's a brain slice here, and it's got a bunch of neurons at different layers of the brain. At the bottom layer of the neurons here, we've managed to fill all of these neurons with a calcium-sensitive dye. And if you were to record electrically from this cell here, when you stimulated the cell, you'd see some kind of pattern of action potentials occurring within that cell. These would be intracellular recordings, because you notice they only go one way, they go up. So those are intracellular action potentials. But we don't have to stick an electrode into that cell. What we can do is measure the calcium, the fluorescence of the calcium dye, and each time there's an action potential, shown here, there is a little increase in the calcium concentration in the cell. A little calcium comes in with the action potential through voltage-gated calcium channels. And so you can sort of take this signal here and use a computer program to basically guess where the action potentials occurred by looking at the inflections in the signal. Okay, and so that way you can get an impression of the activity of this neuron. Yeah?
On the previous slide, is there a reason why it's excitatory, inhibitory?
Yeah, there is. There is.
Why isn't it just excitatory and then slowly?
We'll get to that later. It turns out that it's enormously important for sharpening up the perception of where things are. Okay? It's called lateral inhibition. We'll talk about it mostly in the visual system. So here's a recording here that is made now of a whole group of with these calcium-sensitive indicators, and we're looking down on the surface of a tissue, actually a part of the olfactory bulb in the brain, so it's part of the brain that responds to smell signals. And so this is the part of the brain that we're looking at here, and you can see perhaps all of the little neurons that are there. Perhaps we can... I can't get it. Maybe I can lower the lights here. Yeah. You can see perhaps the outline of the neuronal, some of the neuron and glial cell bodies here. And each of these black spots here represents a little clump of neurons that is being activated during the presentation of an odor. So we're looking down at the fluorescence, and where it's dark, that's where there's more fluorescence. I apologize for that, it's just the way it was coded by the computer. So if you give a smell, present a smell to the animal's nose that smells like a pineapple, okay, then this little group of neurons here and this little group of neurons fluoresce. When you present an odor that smells like a banana, then a sort of somewhat different pattern of activity emerges. All right? And so the advantage of using the fluorescent dyes is that you can sort of get a rough image of which neurons are active and which ones aren't, and get an idea of the complexity of the task that the brain has to do to distinguish between pineapple and a banana. Yeah?
Are you able to resolve individual cells in there or just regions?
No, you can train the microscope down onto an individual cell, and then you'll get a recording like this if you're lucky, and you can kind of try and deconvolve when the action potentials occur. All right? So the main practitioners of this technique in our department in biology are Patrick Connaught and Ricardo Araneda. Are using this sort of approach, all right? So, and I've done a lot of this stuff as well in the past, but on single cells, not on brain slices. All right. Okay, so those are invasive methods, and there's also a bunch of non-invasive methods. Oh, we've got so much time left. All right, so we're going to run through these, because I don't want to leave stones unturned. This is a 300 level course, so we should be able to bite the bullet and learn about the details. So the first thing about these methods is these are all methods to look at gross brain function and anatomy without invading, right? Unlike the previous methods. And the first thing to understand about them is which methods are give you purely structural, anatomical information, and which methods tell you about function. Okay, and the other one is to have a sort of broad idea of how they work. And I hope there aren't too many questions from physicists, because I will probably not be able to answer them. They'll put them on a discussion board and have a crack. Okay. So, the one I'm most comfortable with is computerized tomography, because this is essentially simply a computational technique. It uses, the basis of it is just X-ray images of the brain, much like the X-ray images of any other part of the body. But the X-ray images are taken from a whole variety of angles and perspectives, and basically a computer program is used to reconstruct a 3D image. And there's a lot of fuss, actually, when the people who invented computerized tomography, the electrical engineers who invented it, were given, I think, a Nobel Prize. There's a lot of fuss from other scientists, because they said this is just technical. This is not actually an advance in scientific thinking. So it shows you how bitchy scientists can get. Anyway, so that's computerized tomography, and of course this is just structural. All right, and there's a lot of controversy about its use because the X-rays increase the chances of mutations occurring in the DNA of cells, and so increase perhaps the cancer risk. So there's a lot of controversy about how often people should be subjected to CT scans. So here you might have done this. I know I've done it once. Not my head. Somewhere in the middle here, I think. And you basically take an X-ray source and rotate it around the patient and move it up and down and reconstruct the three-dimensional image here. So this can tell you about, really, what it tells you is the sort of information about how tissues absorb X-rays, which parts of the tissue absorb X-rays and which ones don't. And it turns out that different areas within the brain will absorb X-rays more or less. In this case here, you're seeing the distinction in a cross-section of the brain. These are the ventricles here, so there's just liquid inside of those. Out here is mostly nerve fibers. This is the so-called white matter of the brain. And then out here is the grey matter, where most of the neuronal cell bodies are. So we're looking at a section here through the cerebral cortex, sort of a section that takes, if you cut your head open like an egg slice, or if you were slicing an egg in an egg cup, that's what it would look, that's what you would see, all right? Section through your cerebral cortex here. Okay, so then there's magnetic resonance imaging. And what happens with magnetic resonance imaging is that the hydrogen nukes, the spinning hydrogen nuclei in water molecules are aligned in your brain by a very intense magnetic field. So you line up, you use a very intense magnetic field to persuade all of the spinning water molecules to orient in the same direction. And then you irradiate the water molecules of the brain with a brief radio frequency pulse. And what happens is that the water molecules will resonate, their energy will resonate with the radio frequency pulse. And you can measure the degree to which energy is absorbed or released following the pulse. And so the image is therefore based on the distribution of water in different tissues. That's essentially what an MRI measures, is the distribution of water in different tissues. And the characteristics of the energy emitted by hydrogen nuclei in different tissues, outside of water, other hydrogen nuclei. Okay, so that's essentially what's being measured. It has a high resolution, like CT, the X-ray technique. That is, it has a resolution of less than a millimeter or so, which, as you can imagine, is not a high resolution compared with some of these other imaging techniques, these microscopic imaging techniques. There are literally hundreds and hundreds of thousands of neurons in every cubic millimeter of your brain. So you're not going to be able to distinguish much in terms of individual neuron activity at all. But it's called a high resolution, spatial resolution. And again, it's anatomical and not functional. It's just telling you basically how much water there is, or what's the status of the hydrogen nuclei outside of the water. So, as you know, the magnetic field is produced by a giant circular magnet, which sort of constrains its use. Now, it turns out that brain tumors have very different, contain very different amounts of water from other healthy brain tissue. And so you can actually pick up tumors quite readily using the MRI technique. And that's shown here. Here's a brain tumor here in the middle of the cerebral cortex in this case. Now, there's a variation of MRI called functional MRI. And we have one of those machines on campus, arrived last year. So we're now fully equipped to do research, and some of you might like to be subjects. Go to the NACS website, the Neuroscience and Cognitive Science website, and you'll see links to the imaging center. And you'll see whether they need volunteers. Normally they do it all on themselves, or graduate students. But there may be some projects, and you can always get hold of the researchers and email them and see if they've got any internships going. So the fMRI, what it does is it turns out that hemoglobin slightly distorts the MRI resonance properties of nearby hydrogen nuclei. So there's a little bit of a shift in the resonance of the hydrogen nuclei when they're hit by that radio frequency pulse. And it turns out that the degree of distortion depends on whether hemoglobin is loaded with oxygen or not. So what fMRI measures is the amount of, what it can measure is a signal that's related to the amount of hemoglobin with oxygen bound to it. Okay. Now, it turns out that in active areas of the brain, that is, areas that are where the neurons are generating a lot of action potentials, there's usually an increase in blood flow, which is sort of related to that communication between neurons and glial cells that we talked about. And that blood flow, that increased blood flow, results in more oxygen-rich hemoglobin being supplied to the active neurons, which makes perfect sense, right? You don't make action potentials for free. You need to recover all the ions after the action potential. That costs ATP. So what fMRI measures, it's really what it infers, the measurements infer, are changes in the blood supply to different parts of the brain. And this results immediately in some limitations. Firstly, it takes seconds for the blood supply through the microvasculature to increase in response to activity. And so this is a relatively slow measurement. And secondly, secondly, it is quite spatially accurate because it's a derivative of MRI, so it's accurate to within a millimeter, or perhaps a bit less. As I said, but it's slow. And it's indirect, right? It's not going to tell you what kind of computations neurons are performing. It's just going to tell you whether one area in the brain is active or not. And so when you read about MRI studies, and we look at them later in the course, you have to, remember these limitations. Yes?
What do you mean by slow and why?
Well, because the vasculature, what happens is the blood vessels have to change their diameter, or the muscles have to relax, or the micro blood vessels, and then new blood has to come in, and it takes many seconds for that to happen. So I guess the time scale of seconds is slow, right? Because an action potential happens in a millisecond. And you, for instance, a reflex is all over and done with in a fast one within a couple of hundred milliseconds. So it's not going to be able to tell you what's going on a moment-to-moment basis anywhere. Yeah?
I mean, like, does slowness really have an effect? Because if the delay is always constant, then you know how much the delay is by.
No, it's because what will happen is you give a stimulus, right, and you'll get something that looks like that with a time scale of seconds, right? Like a change of that, like that. And so you can't really tell what's happening within that time scale. Yeah? It's a bit like here with the calcium imaging. You're trying to deconvolve the inflections in this signal into action when the action potentials occur. And there just comes a point when if the signals are slow, the inflections get less and less obvious, and it becomes impossible to do it. Right? And that's the case with MRI. Here, the calcium signal rises, in a few hundred milliseconds, or in tens or hundreds of milliseconds. Milliseconds, not seconds. So when you participate in an fMRI study, it's really boring. because they make you do the same task again and again and again and again, so that they can build up an image of several seconds, over many seconds. Yeah. All right. Now, there's a different method of doing this functional imaging on a slow time scale, which is somewhat similar to the MRI in terms of what it produces, but very different in terms of the way it works. And it's called the PET scan, positron emission tomography scan. And basically what happens is the patients are injected with radio-labeled glucose or oxygen. All right, so let's take glucose as our example. They would be injected with glucose that contains 11C rather than 12C. And this is injected into the bloodstream. Glucose will cross the blood-brain barrier, as will oxygen, of course, and go into the brain. And you detect, what happens is when these guys decay, they emit positrons. And so you detect the emitted positrons with scanning detectors, and you can reconstruct the location and density of the labeled oxygen and glucose. And so, again, active areas will show increased presence of the labeled oxygen, or the labeled glucose will accumulate because the blood flow to those regions is increased. Okay, so it can show you active areas. So the outcome is very similar to what you get with the fMRI, out of the fMRI, but the technique is different. It has a poor resolution, relatively poor, that is a resolution of a few millimeters. And as I understand it, it needs an on-site cyclotron or access to, the positron-emitting compounds within within a day, certainly. And I think in the States now, a lot of these studies use compounds which are made in Switzerland, I heard recently. That's true, I just put that up and gossip. But they're made in Switzerland and then flown to the States for use here. So that's another problem. And of course, I think it's a lot more difficult to persuade a patient to ingest radionuclides than it is to get them to do an fMRI scan. But hell, I don't know. Never been in that situation. Okay. So this is a PET scan of somebody who has trying to perform a face matching exercise. They're given a couple of faces and they have to remember which face is which. And here it is, and you can see this little part of the brain down here lights up during the face matching exercise. Yeah.
The previous picture entirely PET, or was it PET overlaid?
I'm sorry, yes, this is PET overlaid with I'm sorry, I didn't mention that. The PET scan is overlaid onto probably MRI scan. Yeah. So the last technique is, or the second to last, is electroencephalography, or EEG. And so with this technique, a bunch of electrodes are placed onto the scalp of the individual, and plenty of people do this on campus. You sort of glue them on with conductive jelly, or you have this sort of hairnet that contains all these electrodes. And you measure the electrical activity on the surface of the scalp. You're measuring the currents that are flowing along the surface of the scalp in much the sort of same way, but on a macro scale. And the hope is that those currents reflect changes in the underlying activity of neurons, or glia, or both. All right, so as the activity of the neurons changes, so the currents change. And again, you use sophisticated computer programs to take the information from each of the electrodes, and there may be as many as 60 or so electrodes, and try and reconstruct where the signals originate, where the currents originated from in the brain and how they flowed, in order to look at differences in activity. So this can be used to detect activity in gross areas of the cortex, large pieces of the cortex, for instance, because that's the part of the brain that's closest to the skull. And it's got increasingly sophisticated in terms of its ability to pinpoint where these changes are occurring. But the question is, where does the signal come from? Does it come from neurons? Does it come from glia? And what does it represent in terms of actual computational processing? So most of these tools we've talked about are really good diagnostic tools. For instance, the electro and EEG recordings during sleep are very different as somebody goes into from a wake state to deep sleep. The patterns of these EEG recordings from these electrodes varies very characteristically. But exactly what this tells us about what the brain is processing during sleep or doing during sleep is really obscure. But if you want to know whether somebody has a sleep defect, whether they're able to dream, if they're exhausted during the day and they're not able to go into deep sleep, you can run an EEG and use it diagnostically to see how deep their sleep is. The last and most sort of impressive piece of technology that's used non-invasively is called SQUID, or magnetoencephalography, MEG. And instead of using a sort of hairnet of electrodes on the scalp, what the SQUID apparatus involves is it uses superconducting magnetic detectors, superconducting quantum interference devices, that's where the SQUID comes from. And these detectors that detect magnetic fields are placed around the patient's head, and they measure magnetic fields within the head. And again, by placing a whole bunch of these detectors around the head, you can use computer programs to reconstruct where magnetic fields change within the brain. And remember that when current flows around a circuit, it generates a change in magnetic field as well. And so again, what you're measuring here are gross changes in current occurring, flowing around neurons and glia of fairly uncertain origin. But the beauty of this technique is that it is able to localize these changes to within a centimeter or so, which is much better than the EEG can do. And it has millisecond resolution. So it can measure very rapid changes in the gross activity of a fairly large piece of brain. None of the other techniques we've talked about have this sort of combination of speed and spatial resolution that SQUID has. And we have a SQUID device also, oddly enough, belongs to the linguistics department. Although I think the manager is Jonathan Simon, who's in biology. So we have one of these, and again, I think they need to look them up on the NACS website. Maybe they need people. And it looks like looks like that. Sometimes it looks like a little giant hair dryer. But again, the problem is that you've got to do this and then perform some kind of task at the same time, and you've got to do the task again and again and again so they can average the traces and try and reconstruct the magnetic field activity. And so it's really boring, but great fun. Okay. But you get, this image, go home and say, look, Mom, this is what was going on inside my brain in college. This is what you're talking about. Okay, so I'll see you on Tuesday, and we'll rush through some very basic anatomy.